right, members, I don't know what had happened. Someone was trying to hack into the system. I really don't know. Or maybe the internet connection was poor. I really can't tell. Good enough, we are here. Let me see. There's some members who left. I don't know what happened on your side. Were you logged off automatically?
Yeah.
Teacher, you were off, but for us, you were fit.
You were on.
No, teacher,
Yes.
we were not on. Some of us.
Okay. Most of us were on. It's only you and maybe Petronella.
Okay, I think
Even
it was
Bakita.
network connectivity. Yes.
Even Bakita was off.
Okay, so everyone was off. I thank God that we are all back. What was I talking about? Can someone remind me?
DNA. No, protein synthesis.
Uh-huh.
No, teacher.
Yes.
Teacher,
Yes.
excuse me, sir.
You are
Okay. You left a question pending. You are like, should we start from DNF or from protein synthesis?
Yes. And what is your
Yeah.
answer?
Namanda was like synthesis, but for us, okay, for me, we start from DNA.
DNA, which we have already started, right?
Yeah.
Come again?
Yeah, to go.
I'm hearing you in slow motion. I don't know whether the rest are also hearing you in slow motion.
I really don't know. Okay, I'm like, let's continue with DNA.
All right. So,
Yeah.
I wanted to proceed on to protein synthesis, but before I proceed to protein synthesis, there is a video that we watched, that song that we watched, right? I want us to give some of the characteristics or to describe the structure of the DNA. Can everyone unmute and you tell us at least one part that makes up the structure of the DNA. A description of the structure. Yes, Amanda, beginning with you.
It is made up of a deoxyribose pentose sugar.
Very good. The pentose sugar is deoxyribose. Let me see here. Deoxy. Our charts also disappeared. I think we shall keep on reminding ourselves. It's made up of deoxyribose sugar. It's a pentose sugar. That's very good, Amanda. Can I have someone else? What else makes up the structure of the DNA? Nabuzare.
Yes, teacher.
Yes, talk to us.
Yeah. It's also made up of nitrogenous bases, that is purines and then pyrimidines.
Okay, it's made up of the purines and the pyrimidines. Can you give some of the examples in each case?
In purines, we have adenine and then guanine.
Yes.
Then in pyrimidines, we have cytosines and then thymine.
Yes, cytosine and thymine or thiamine. Okay, that's very good. Catherine, I don't know how I can clap for you in this meeting, but look for the claps and I clap for you. Let me see. Yes. Okay, can we have someone else? Yes, Betty Akumu has clapped, has seen the claps. Betty Akumu, what else do you have to say? Yes, we can hear you, Betty.
Teacher,
Yes.
the DNA is made up of the hydrogen bonds.
Okay, it's made up of the hydrogen bonds. Where are they found exactly? Can we be more specific?
Between the nitrogenous bases.
Okay, so it is made up of hydrogen bonds that hold on the purines and the pyrimidines together, whereby guanine and cytosine bond via a triple bond, triple hydrogen bond, whereas adenine and thymine via a double hydrogen bond. That's very good. Who else? Who else has not said something? Let me look for the participants here. Here you cannot hide, you must talk. Huh? Betty. Morris Mwanje, you are so quiet. Talk to us. Remember, we talked about yesterday, we mentioned something. And I'm preparing a present for whoever is going to be an active participant throughout all the sessions of biology. The most active participant shall be awarded.
Teacher,
Yes.
it is made up of the phosphoric acid.
Okay. Or we can call it a phosphate group. That's good. Phosphate group that is bonded to the nitrogenous base. So it is made up of a sugar phosphate backbone. That's very good, Mwanje. Thank you for your contribution. I'm seeing a hand. Whose hand is that? Yes, Petronella.
Double stranded.
Yes, it is made up. It is a double-stranded polymer. That's very good to highlight.
Double stranded, highly folded.
Come again.
Double stranded and highly folded.
That latter point, that latter statement is making us not accept your answer, but we can say it is has a double-stranded helical structure, or it's a double helix. Is that clear?
Yes, but
Because
teacher, it's highly folded as well. Why is it wrong to say that it's not, that it's highly folded? Because even in all the books they say it's folded and you can stretch it for a long distance.
okay, I think I thank you for that idea. However, when we say it is highly folded, a folding is a disorganized form of arrangement. It's a disorganized structure. And that folding, for the DNA, its coiling, it's like that of a spring. Have you ever seen a spring?
Nope, I don't even know what that is.
In physics, in O-level physics, we're using some springs that could stretch.
A screen, a what now? Teacher, first repeat what you said.
It is a spring. Let me type it in the chart. The spelling is a spring. Have you seen that?
Oh, yes, I remember that spring. It was like Hooke's law.
Okay. Yes, so that would be the DNA structure. Rather, the shape of the DNA. So it is a double-stranded. It has a double-stranded helical structure. And the structures, I think you can see it on the screen, right?
Yes. So now,
Okay.
teacher, do you
Yes.
see what I'm meaning? Like those foldings, there's a way it forms something like 8.
Okay, so that shape, we call it a helical structure.
Oh, right.
Yeah, that is the name given to that shape. It's called a helical structure.
Okay, teacher, just imagine if DNA was like just a ladder, like now each structure is just like 2 lines bonded by hydrogen. How would you call that shape?
Come again.
I'm saying, what if like DNA,
Mm.
okay, that part where it's unwinding, like it's just like a ladder.
Mm.
So how do we call that shape?
You've already mentioned the shape. We can simply call it a ladder shape. Mm. You've mentioned the shape. So it is a ladder shape, or you can call it a spiral. Actually, it's a double helix that can either be having a ladder shape or appear like a twisted ladder or a spiral staircase.
Okay.
Okay. I hope we are there. So that is good, Petronella. Thank you for bringing up that discussion. Yes. Then I've not heard someone mentioning that the structure is anti-parallel. The two strands are anti-parallel. You'd appreciate that, I think. Who else has not spoken? Who is maybe Mr. Sentai wants to say something. Let me make him a co-host. Mr. Moses.
Good morning, everyone.
Good morning.
Uh, are people hearing? I've just come.
Yes.
Yeah, and I can see the lesson is going on well. Yeah, that's all. I'm just here. There's nothing much I can say.
You
Let
are just
people continue learning and appreciating the science.
okay. I think you'll help me administer the charts if possible. I don't know whether you'll be logging out soon. Okay, so we are continuing, members. We are continuing. We mentioned that DNA is made up of it's made up of nucleotides. Actually, it's a polynucleotide that is made up of nucleotides, and each nucleotide is made up of a pentose sugar and a phosphate backbone. Then on the pentose sugar, there is a nitrogenous base that is covalently bonded to it. Then it's a double-stranded polymer, and each strand runs in the opposite direction, or we can call that it being anti-parallel when the two strands are running in the opposite directions. Then Petronella was mentioning that it has yes, it is a double strand that has a double helix shape, which we corrected, I think. Yes. And I don't know whether someone else has something else to say before we go on to protein synthesis. You should take note that after this lesson we shall have an assignment that shall be attempted and done online. I'll have to mark that assignment. So as we are learning here, make sure in case you have something you've not understood, you ask a question. You can either type it in the chat or raise up your hand. We ask you to unmute, then ask the question. Any other question regarding the DNA? In the next 10 seconds, we shall be looking at protein synthesis if there is no question. Remember, we are going to watch a video and From this video, I'll have to ask you questions which you've to tell us what you've learned from the video. Then we proceed by harmonizing our responses. Okay, we shall have 4 different videos combined, and I think by the end of the 4 videos we shall have Here goes the first video.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Let's look at the second video.
Protein synthesis, translation. Inside the body, the process of translation occurs within every single cell. Each cell has a nucleus. After transcription, mRNAs move out of the nucleus and enter the cytoplasm. This mRNA strand acts as a template for protein synthesis. Present in the cytoplasm is an enzyme, aminoacyl tRNA synthetase. The enzyme macromolecule has 2 binding sites. One site recognizes the amino acid methionine. This is followed by the binding of the ATP molecule and release of pyrophosphate, resulting in activation of amino acid. Finally, the tRNA and the activated amino acid bind together. This amino-acylated tRNA is known as met-tRNA and is released from the enzyme. This marks the commencement of first stage of protein synthesis, the initiation stage. During the initiation stage, a small subunit of a ribosome binds to the mRNA strand. The mRNA strand is made up of codons, which are sequences of 3 bases. Then the ribosome subunit moves along the mRNA in 5' to 3' direction until it recognizes the AUG codon or the initiation codon. At this point, MET-tRNA possessing the anticodon UAC pairs up with the AUG codon of the mRNA. Then a large subunit of ribosome combines with a small ribosomal subunit. The large subunit shows 3 sites: the acceptor site or the A site, the peptidyl site or the P site, the exit site or the E site. This whole unit forms the initiation complex. This is followed by the elongation stage. During this stage, another tRNA carrying molecule of an amino acid approaches the mRNA ribosome complex and fits in the A site. Then a bond is formed between methionine and the amino acid molecule on the tRNA. As a result, MET-tRNA becomes deacylated. The ribosome then advances a distance of 1 codon and the deacylated tRNA shifts to the E site from where it dissociates. Meanwhile, another tRNA carrying an amino acid molecule attaches to the A site. This is followed by the binding of the amino acid molecules. Repetition of this process leads to the formation of an amino acid chain. This event is called elongation. Finally, when the UAG codon or the stop codon reaches the A site, elongation is terminated. Termination is the last stage of protein synthesis. The chain of amino acid molecules is released from the ribosome. This released amino acid chain is the protein, and this part of protein synthesis is known as translation. Then the tRNA detaches from the mRNA. Ribosome detaches and dissociates into its small and large subunits. Summary. Protein synthesis shows that the first stage involves the binding of MET-tRNA to mRNA and the small subunit of the ribosome. The larger subunit of ribosome then combines with the small subunit. Second stage is the elongation stage. In this stage, the incoming amino acyl tRNA fits in the A site. Then a bond is formed between methionine and the amino acid molecule on the tRNA. The process is repeated until a chain of amino acid molecules is formed. The last stage of protein synthesis is the termination stage. When the ribosome reaches the stop codon UAG, elongation stops and the newly formed amino acid chain, which is the protein macromolecule, detaches from the ribosome. Subsequently, ribosomal subunits along with the tRNA dissociate from the mRNA.
Here's the third video.
For our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. Where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. It begins with a promoter and ends in a terminator. Also contain regulatory sequences that can be found near the promoter or at a more distant location. In genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. Transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. Process occurs in 3 stages: initiation, elongation, and termination. Initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. This causes the DNA double helix to unwind and open. During elongation, the RNA polymerase slides along the template DNA strand. Complementary bases pair up. The RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The end of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. For the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. The process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. Complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? Nitrogenous bases are grouped into 3 letter codes called codons. Genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Amino acid is brought to the ribosome by a specific transfer RNA molecule. Type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. The large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. In elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. The anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. Complex slides down 1 codon to the right where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Propagation will continue until a stop codon is reached. This factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. Their complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. For dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. See how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA.
Okay, someone is asking for the soft copy of the textbooks before you even finish the video. I think I'll have to send. Thank you for the reminder. Thank you for the reminder. By today, I promise I'll send. Amanda Glory, I hope you are sorted.
Which then passes through the ribosome like a computer tape. Amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. A small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
All right. right, I think that is it for now. We've seen 4 different videos speaking about the same thing, and I believe you've been learning something from each of the individual videos. Before I ask you questions, I want you people to unmute and ask questions and seek clarity where needs be. Yes, Catherine.
Yes, teacher. They talked about extrons and introns, but I didn't understand because it was somehow fast.
Okay, exons and introns. Okay, that is according to Catherine. I don't know whether someone can help me type in the chat. I was looking for it to note, but my notebook is far. Can someone type in the chat? Or I can, maybe Mr. Center if he's around. Exons and introns. Yes, who else? Amanda? Petronella, you are unmute. You have unmuted. Talk to us.
Yes. My problem is there where the chain starts elongating. Okay, how to explain that part, the part of the ribosomes coming and the large and the small? I'm having a problem there.
Yeah, so basically the process of translation, right?
Yeah, yes, teacher.
Okay, that is
so the
yes.
P sites and E
Yes.
sites and S sites. What are those things?
AP and E sites. Okay, let me type them here.
Master, me to my concern is like for that, for Gloria.
Gloria's concern. Exons and introns.
The translation there. Yes?
Translation. Is that what you're saying?
Yeah. Yes, teacher.
Okay. Any other person? All right. It seems those are the most challenging parts. The others, maybe they are still looking for the buttons to unmute. That's my assumption, but I believe you know how to unmute. All right. Let's begin with the first concern: exons and introns. Now, let's take an example of insulin, which is a hormone protein in nature that is secreted by the pancreas, the beta cells of the islets of Langerhans in the pancreas. So, inside the cells of the pancreas, the beta cells of the pancreas, each beta cell has a nucleus. And that nucleus has chromosomes. Remember, if it is a human cell, if this cell is extracted from a human being, it will have 23 pairs of chromosomes. And on each chromosome, there are sequences of genes. There are sequences of genes on each chromosome. Now, in case the gene that is responsible for secretion of insulin is found near other genes. For example, the genes that are responsible for the hair color, the genes that are responsible for the blood group or blood type, the genes that are responsible for your skin color, and so on. in case the gene for secretion of the insulin hormone is found in between those other genes, where the initiation site, I'm speaking of a lot of biology, and I'm very sorry for speaking a lot of biology. You'll have to bear with me. Now, let me get an image here that I'll use so that it becomes my teaching aid here. Let me get this image here. Get an image here, just a second. I think that image will allow us understand better. Which one should I select? Okay, let me choose this one here. Okay, I think here's the image. Can we all see that image on the screen? If you can see it, raise up your hand. Yeah, Nabuzali has seen the image, and Adikini. If they have seen the image, it means that the rest have also seen that image. So this image is showing a section of a chromosome where genes are found. Okay? Before transcription occurs, the RNA polymerase enzyme must bind to this red region first, which we are calling the promoter region. Then, along where the promoter region stops, it's where the transcription site starts. Where we have, from the previous video, we saw the AUG codon that codes, rather the AUG code that codes for amino acid methionine. Namatov has just joined. Yes, Namatovu, you are welcome, and we have about 50 minutes. Thank you for joining very early. So, back to my description. I was mentioning that the RNA polymerase enzyme binds at the promoter region. Then, along this transcription unit, we find different codes or different genes that are supposed to be expressed. Then here it's where the RNA polymerase stops when it reaches the termination region. Now, along the transcription unit, there are several genes. Just like I earlier mentioned, we could have a gene for hair color, we could have a gene for skin color, we could have another gene for secretion of insulin hormone. But since rather insulin is secreted when the levels of glucose in blood have risen, meaning rise in glucose levels be above optimum or above threshold, stimulates protein synthesis or synthesis of insulin hormone, and it begins from the DNA. So, and in case the gene for insulin is found in between that RNA polymerase cannot jump the other genes that it is looking for insulin alone. What it will do during transcription, it will carry out transcription of all the other genes that are found where the insulin gene is found. And so after transcription has occurred, this messenger RNA that has been formed must undergo processing. Remember, our interest is to secrete the insulin hormone. The other genes for hair color and skin color are not needed at the moment. So what happens during processing of the mRNA, the messenger RNA, specific enzymes catalyze the removal of these unwanted genes or codons that contain the unwanted genes. That process is called splicing. Let me write it in the comment section. Splicing. Someone is hearing echoes. Amanda, who else is hearing echoes? Members, raise up your hand if echoes are being heard. Oh, sorry, let me take this gadget away. Oh, I mute it. Are echoes still being heard? Do you still hear the echoes? Raise up your hand if you are not hearing the echoes. Okay, I think now it's clear, Amanda. No more echoes. Thank God and thank you so much. So I was going to the comment section to type in the spelling of the word splicing. There it is. And that process of splicing or cutting off the unwanted genes or codons is called splicing. It is catalyzed by a group of enzymes that are called spliceosomes. This is for purposes of you to understand, but if you want to learn more about the spliceosomes, you'll find that those concepts at university level. In case you want to learn more about the splicing and the spliceosomes and mRNA processing in details. But just for purposes of understanding, the messenger RNA that has been transcribed undergoes the process of splicing, which involves removal of sections that are unwanted, which we call the introns. Introns, from the word inside. Introns remain inside the nucleus. They do not leave the nucleus. We are talking about eukaryotic cells. However, for prokaryotic cells, the prokaryotic cells don't have introns and exons. They only have exons. They don't have introns. Then, the sections that exit the nucleus and go to the nucleus for translation to occur, they are called the exons. They are called the exons. If we are together up to there, up to that point, raise up your hand. If we have not raised up your hand, it means that we are not together, and we shall ask you to tell us what we should repeat. Okay. Leave your hands raised up, please, in case we are together up to there. Amanda, Navzale, Petronella, rather, yes. Then Pavintendo, we are together. Then the rest, we are not together. That is what it means. Elizabeth Achen, talk to us. Why are we not together, Achen? Bukenya, Ibrahim has sent it in the chat. So we are together with Bukenya. Elizabeth, why are we not together? Unmute and talk to us. Before also Namatov Maria also tells us. Then also, please, those people who had raised up their hands, keep your hands up. I want to see those people whom we are not together with. A guest, okay. Betty Akumu, we are not together. I don't know why. Please unmute and talk to us. What is not happening? Am I so fast? Am I so slow? Am I talking about complex terms that you are hearing for the first time?
Okay, teacher.
Yes.
I'm requesting you to repeat.
What should I
like
repeat?
Everything.
Come again. Betty Akumu, I didn't hear you very well. What we should repeat? Unmute and talk to us. Let me ask you to unmute. Teacher, we are cooking lunch. Petronella, you are cooking lunch. Should we take you to the waiting room so that you cook lunch as we wait for you to finish cooking lunch?
No, teacher, there's no problem.
So how are you writing it down as you're cooking lunch?
I have many hands.
How many?
2 hands.
2 hands. Everyone has 2 hands unless there's someone who is here and they are handicapped.
No, teacher, we are just good at multitasking.
Okay. Morris Mwanje, you declined to unmute. I'm going to send you to the waiting room if you are not speaking to us.
Teacher, for me, I'm good. I'm following.
That is very good and very nice to hear. Thank you. Elizabeth Achen has refused to unmute. I'm going to send her to the waiting room. Because at least I want to move with the people who are following. Elizabeth Achen, we are calling you for the last time because I'm looking for the waiting room. I'm going to put you in the waiting room for some minutes. Then I bring you back. Teacher, it is okay because I was not there from the start. What is okay? Maria Namatovu, what is okay and what is not okay? Unmute and talk to us.
Okay, teacher.
Yes.
Teacher.
Yes.
For me, okay, I would have understood, but I was not there from the start. So I'll be disturbing you if I tell you to repeat.
No, your colleagues here, there's people who have been here from the start. I can request them to share with us what they have understood so that I also supplement.
Okay, if they can repeat for me. But if not, it's okay.
Okay, that is Namatov. Who has refused to? Let me send this person to the waiting room because, hey, she has unmuted finally. Talk to us, Elizabeth.
Yeah, teacher, I also joined you guys late. There's the same story like Maria, but I'm trying to catch up.
You're trying to catch up. Okay. Because I was looking for the button to send you to the waiting room, but you have survived. Thank God. The Holy Spirit is good.
Amen. God is always with me.
Bukenya Ibrahim, I don't know whether you said something. And ultra toxic. Say something, ultra toxic, as we invite Pavintendo to basically tell us what you have understood in your own words, the way you've understood. It is not a do or die. Just tell us what you have understood. Yes, ultra toxic, talk to us as we wait for Pavintendo.
Yes, teacher. My network was a little bit poor. This some minutes I was off.
Okay, so you missed that point. It means that someone has to repeat for you, or we shall send for you the recordings and you review. Yes, Pavintendo, we are waiting for you. We can't wait to hear your voice. Please tell us what you've picked from the session that I mentioned, I talked about earlier on.
So I have talked about the exons and introns.
Yes.
You said the introns are not really necessary, so they remain in the nucleus.
Okay.
And then the exons are the ones that are necessary during the process, so they leave the nucleus. Yes, that's what I've understood.
Okay, that's very good. Hands, I think I should clap for you. Where are the hands? I'm failing to see my hands this side. Someone should clap for Pavintendo. Then Amanda Gloria, tell us something. Yes, Petronella, thank you for clapping, and Nabuzari.
So teacher, you've been talking about insulin, formation
Yes.
of insulin.
Yes.
Then you said that insulin is formed by the beta cells
Yes.
in the pancreas.
Correct.
So you said that in the DNA to code, okay, the DNA to code for the formation of the insulin, there may be some introns and extrons. Now the introns are the unnecessary parts that remain inside the nucleus. They may be genes that are coding for hair color. What else? Okay, they may be coding for other characteristics, not insulin. So those are the introns. Then the ones that move out of the nucleus that code for the insulin are called extrons. They move out of the nucleus into the cytoplasm. That's what I've understood.
Okay, I think we should clap for her. That's very good. You've been so attentive. Let me clap for you physically now. Yes. Maurice Mwanje has clapped for you. Petronella wants to say something. Unmute and talk to us, Petronella.
Okay, teacher, I have a question here.
Yes.
Still about like that. Okay, it was something connected to the video.
Yes.
Some other, I think the third or fourth video. They talked of something like when the protein has been, the amino acid has been formed, it now goes into the what? The Golgi apparatus. No, not the Golgi apparatus. Okay, I've forgotten where it goes, but it goes into something and then it also goes through the Golgi apparatus. So when you're answering that question, is that necessary to put, like answering the question of how proteins are formed? Must you talk about the Golgi apparatus and then talk about like the the rough endoplasmic reticulum? Yeah.
Okay, if the task is direct and clear, and they are asking you to describe the process of protein synthesis. You begin right from the DNA and end in translation, the process of translation. That is transcription, processing of the mRNA, then translation. However, in case the scenario is talking about secretion of a given substance which is protein in nature, it means that you have to begin from the nucleus, move to the cytoplasm where translation occurs, that is on the rough endoplasmic reticulum. Then from the rough endoplasmic reticulum, this crude protein, I'm saying crude in quotes, the crude protein is packaged in vesicles and transported to the Golgi apparatus, and the role of Golgi apparatus is to label this protein here, is to add markers on it and label it, because this protein it might go and function in another part of the body. So in case you are a person and you are sending a message to someone, before you send the message you must have their phone number or WhatsApp number. So that WhatsApp number is a label that tags that message that is going to this person. So in the same case, the Golgi apparatus carries out processing of this protein, where it changes the primary and the secondary structure of the protein into the tertiary and quaternary structure, and also add on markers that label it and package the protein then into secretory vesicles. Then this protein here is secreted by exocytosis. For the case of insulin, insulin is a hormone, and hormones are produced by endocrine glands, meaning as you're describing the process of formation and secretion of insulin, after mentioning of the Golgi apparatus packaging into the vesicles, then the endocrine glands have cells that have red blood, rather that have blood vessels within or in their very close proximity. So instead of mentioning it's being secreted by exocytosis, it's that they are secreted by exocytosis into the blood vessels, because insulin is secreted directly into the blood vessel. I think that is what I can say to you. That is Petronella. Is that clear?
Yes, teacher. It's very clear.
Okay, that's good to hear. Someone else had something to say. Okay, let's go back to we are talking about the exons and introns, and that is very clear. Then someone mentioned that we should talk about translation. Now, translation can be categorized into two sub-stages. Betty Akumu had left us and is just also in the waiting room. I don't know whether they were buying data or they were activating what. Yes, Betty Akumu, we are glad that you are here with us with Catherine. You had left us. I don't know whether you had joined the discussion room. But here we want to talk about translation. I was mentioning that translation can be categorized into two sub-stages, or whatever events that are occurring in the cytoplasm that cause protein synthesis to occur can be categorized into two. One is amino acid activation. If you are writing, please do write. Amino acid activation. It is a two-step process. Amino acid activation. During this process, remember in the cytoplasm, we have amino acids that are there in abundance, in addition to tRNAs, transfer RNAs that are there but also in abundance. However, they have to be mobilized and arranged in a way that they will form a polypeptide chain that is corresponding to a protein that the body needs at that particular moment. So before assembly of these amino acid occurs during translation, amino acid activation must occur. And during this this stage here, we have what the amino acid activation can also be called amino acylation. Amino acylation. It's also another name for amino acid activation. So during this process, first of all, an amino acid reacts or is bound to an ATP molecule that is in the cytoplasm. Together with that reaction is catalyzed by an enzyme, and that enzyme is called amino acyl tRNA synthetase. It's called amino acyl tRNA synthetase, and that is the enzyme that catalyzes the process of amino acylation. Just to re-echo this, I mentioned during amino acylation, the amino acid that is present in the cytoplasm binds to an ATP molecule. By doing that, activation is occurring. Remember, in chemicals of life, ATP is the most used molecule that is used to activate substances. You also remember during respiration, glycolysis, glucose is activated by binding of an ATP molecule to form glucose 6-phosphate. So the same applies this side of amino acylation. The amino acid is activated by binding to an ATP molecule. On binding to an ATP molecule, the substance that is formed, the intermediate that is formed, is called amino acyl AMP. AMP in full stands for adenosine monophosphate. Remember, the ATP molecule has a pentose sugar that is bound to 3 phosphate groups. So during amino acylation, the 2 phosphate groups are replaced by an amino acid to form what we call an amino acyl AMP, adenosine monophosphate. Now, it is this amino acyl AMP that binds to the 3' end of the transfer RNA. Let me get you a picture here of activated transfer RNA. Where are we? Let me use this one here. ScienceDirect.com. Visit. I don't want, okay, let me accept the cookies. Though I don't want to eat cookies now, I want to eat, what else do I want? I want to eat rice, not cookies. All right, here we are. Show more. Yes, exactly. Minimize this. On your right, are we on? Can you all see the picture on your right? Raise up your hand if you can see. Good, good. People are seeing the picture. All right. So here, I don't know whether my cursor is being seen. Okay. I think you can see the 3' end. The 3' end is orange. I don't know whether it is dark orange or orange in color. Then the 5' end is green in color. On the 3' end, it is where we have the activated amino acyl AMP binding. And that is the amino acid binding site on the transfer RNA. I'm giving you some seconds so that you can meditate about what we are talking about, in case things are not going well on your side. You can call the Holy Spirit and it will help you understand more or better. Hope we are there, all of us. Amanda, I hope you are following. Are you following, Amanda?
Teacher.
Yes, Nabuzale, are you also following?
Yes, teacher,
Yes,
I'm
teacher.
following.
Okay, that is very, very, very good to hear. So I can happily proceed. I can happily do what? Proceed. So, the basic steps of amino acid activation, we mentioned that amino acylation or amino acid activation occurs when an amino acid binds to an ATP molecule to form amino acyl AMP. Then the amino acyl AMP is bound to the 3' end of the transfer RNA, and that process is catalyzed by an enzyme called amino acyl tRNA synthetase. So on this transfer RNA, we have this pink part that is at the bottom. That pink part is what is containing the bases that are exposed, ready for translation. So these bases form what we call the anticodon. There are always 3 bases therein. Remember, the genetic code is a triplet code made up of 64 different combinations. To get 64, how we get that 64? It is a simple calculation using some simple math which you don't need to cram. It is very easy to understand. Remember, we have 4 different types of bases, nitrogenous bases. There are 4 different types of nitrogenous bases. But the triplet code is made up of only 3 of those bases. So to get the total number of different combinations of bases that can have codons for the 20 amino acids, we take 4 raised to the power 3. If you press your calculator, 4 raised to the power 3, you'll get 64. I hope that is clear, and you'll try out with your calculator. So 4 raised to the power 63 gives us 64. And that's what gives us the 64 different combinations, the genetic code. The 64 different combinations that are used by DNA during gene expression or protein synthesis. I think in BS and all the other textbooks, you will find a table that is similar to this one here. So if you count UUU up to GGG, all those add up to 64. And in here, we have only 20 different amino acids. We can see different amino acids code. You'd find that an amino acid is coded, one amino acid is coded by 4 different codons. For example, if you take serine or serine, has UCU, UCC, UCA, UGG, UCG. All those is serine. So that Namatovu is taking us back. Let's go back. Namatovu, we shall go back. We are always on your side. Okay? We are always on your side. Namatovu. Namatovu wants us to repeat where ATP binds to amino acid. It's not amino acylation, Namatovu. I mentioned that during the process of amino acylation is the process of activating an amino acid with a tRNA to make it ready for translation. And that activation makes that amino acid charged. Actually, it charges the amino acid. That activation charges the amino acid. And the movement of these amino acids during translation, I don't know whether some of you can wonder what causes, who tells this amino acid that you should move to the ribosome. I don't know whether you've asked yourself that question. However, I'm going to answer you that question, whether you've asked yourself or not. So, during amino acid activation, that process causes the amino acid together with the tRNA to be charged. Remember, from the little physics that you recall in your O-level, in case you have a positive charge and a negative charge, these charges can attract each other. So that attraction is what occurs in the cytoplasm during the process of translation. So, back to amino acylation. I mentioned that the ATP molecule binds to an amino acid that is present in the cytoplasm. And that binding forms an intermediate compound. And that intermediate compound is what we call aminoacyl AMP. That is the intermediate compound. It is this aminoacyl AMP that is bound to the 3' end of the transfer RNA. That process is catalyzed by an enzyme, and this enzyme is called aminoacyl tRNA synthetase. Namatov, raise up your hand if we are together up to that point. Good, that is very good, Namatov. I hope we are now together. Then also with the rest of the individuals, I hope we are together. Biology is biologying here, and I'm seeing time is running out. I wanted us to join discussion rooms. I don't know whether it will be possible, but let us do the needful here, and I make sure at least people benefit here. Sir, type the enzyme. The enzyme was typed here. Let me put a reaction on it. Let me put, which reaction should I use? Let me put an R on it. A pointing finger on it. Where's the pointing fingers? I can't see fingers. Members here. Okay, the finger is there. Bukenya Ibrahim, I want you to be sure. I want to be sure that you've seen the name of the enzyme, aminoacyl tRNA synthetase. Have you seen it? If you have seen it, raise up your hand. Solved, sir. Okay. Thank you. Thank you for that. Thank you. Let's go to yet another part. Someone was talking about the A, P, and E sites shown in the video. Okay. Let me get here an image of a ribosome. During translation. I think this is a better one. This is exactly what I'm looking for. Sir, is that all about translation? Catherine, earlier on I mentioned that translation, we have not yet started the process, Catherine. Translation can be, or whatever happens in the cytoplasm can be categorized into two. One is amino acid activation, which we have just talked about and everyone has understood, I believe. Then the second process is the actual process of translation. And during translation, this is what you have to understand. That's why I brought up this image here of the ribosome. Okay, here we go. During translation, there are 3 or 4 different things that are being involved there. The ribosome has 2 units, a small subunit and a large subunit. That is the ribosome. Then the other thing that is involved is the messenger RNA. That strip of nucleotides that you can see there is what we are calling the messenger RNA. Remember, this messenger RNA was formed during transcription in the nucleus. Then the other thing that we can see here is the activated amino acid. That is a combination of the amino acid and the transfer RNA. The one that was activated during the process that I've just mentioned 5 minutes ago. That process of amino acid activation. So this is the charged transfer RNA or activated amino acid transfer RNA. So back to the A, P, and E sites. On the large subunit of the ribosome, we have 3, I think you can see 3 channels. Let me call them channels in quotes. 3 channels on that large subunit. Then the first channel is what we are calling the A site, or amino acid binding site, or entry site. Then the P is the one that is the channel that is found in the middle. That P site also refers to the polypeptide growing chain site, or in simple terms, it is the channel where the growing polypeptide comes out from. Yes, 8 minutes to go. Thank you for the reminder. You're a very good timekeeper. Yeah, thank you. You'll need a present. That's all about. I don't want to spend much of your time. It is 8 minutes to go, and that is true. So let's finalize by talking about this here. The P is where the growing polypeptide chain or the protein. Where is the first channel? Unfortunately, I don't know whether you can see my cursor on the screen. However, let me use descriptions. where you are seeing an activated amino acid that has a pale green amino acid entering the arrow that is pointing downwards. The place where that activated amino acid is going through is what we are calling the A site or the entry site. Then the P site is this one, which is in the middle, where you are seeing very many, rather you can see an amino acid chain growing from, where you have 2 green circles, 2 red circles, and so on. That is the P site on the ribosome. Then the other side where you are seeing an empty tRNA coming out from without an amino acid, that is the E site or the exit site. I hope that one is clear now. If it is clear, raise up your hand. I need to see whether people are following. Okay, that's very good. If Mwanje says that it is clear, then it means that everyone says that it is clear. Very good. I see very many hands being raised. That's very good. Now, we want to go for lunch, and I'm also very hungry, members. So, during the process of translation, first of all, this small subunit of the ribosome that is found down here first scans the messenger RNA to look for what we call the start codon. The start codon has AUG that codes for methionine. That is what tells the protein synthesis where to start from. Someone is now writing on my screen. Hello. Why are you writing on my screen? You delete what you've written before I send you to the waiting room. Okay. I hope it was accidental, it was not intentional. Maria Namatovu, we don't have the B side. It is A, P, and E site. Let me type them in the chat. A, P, E site. The A site is the one that we have just seen earlier on. Let me look for my pointer. I think people are not seeing my cursor. Let me look for my pointer. Oh, yes. Very good. Let me put it in, use the red pointer. I think, can you see the pointer now? Are you seeing it moving? Raise up your hand if you can see the pointer moving. Good, people are seeing the pointer. Wow, Namatovu, back to you. This one here is the A site. This one here is the P site. And this is the E site. This part here. Once again, A site, P site, E site. All right, let's proceed. Now, I mentioned that during the process of translation, what occurs first is actually there are 3 main stages. The first stage is the initiation of translation, then elongation stage, and the termination stage. During the initiation of translation, the small subunit of the ribosome moves along the messenger RNA, scanning it, looking for where we have the start codon. And once it finds the start codon, it will bind there, and that will mark the initiation stage, the initiation site. And that is the first stage which is done. The elongation site, the elongation stage rather, before we go to the elongation stage, once the subunit has bound to the start codon, then the larger subunit of the ribosome is attracted to the small subunit which is found on the start site or the start codon. Then once it binds there, the unactivated tRNA bearing an amino acid methionine. For all the processes of protein synthesis, methionine is always the first amino acid to start the process of protein synthesis. So methionine marks the start. So methionine bound to a tRNA that has been activated, then to a large subunit, and to a small subunit, and to the start codon, that is AUG of the messenger RNA. All that combination forms what we call the translation initiation complex. Forms what we call the translation initiation complex. That is the small subunit bound to the messenger RNA containing the AUG codon, then which is bound to an tRNA that has been activated with an anticodon that bears an amino acid methionine together with a large ribosomal subunit on the S site of the large ribosomal subunit. That's what makes the translation initiation complex. Now, once the translation initiation complex has been formed, then the ribosome will move 3 bases ahead. In that direction where you're seeing, this direction where we can see this black arrow here. The large ribosomal subunit will move 3 bases ahead. And on moving 3 bases ahead, that is the direction of growing of the polypeptide gene. The ribosomal subunit is moving in this direction. Let me get a better arrow here. Members, you can see the arrow. Yes, I believe you can see the arrow, though it has a different color. Let me put it in red, dark red, so that you see it very well. So the ribosome moves in that direction where you're seeing the red arrow. Then it moves 3 bases ahead, and those 3 bases are what form the codon. 3 bases ahead. Then that shifts the start codon, or rather the start amino acid with its tRNA, onto the P site and creates space on the S site to allow another, here, it creates space on the S site to allow another transfer RNA that bears another amino acid to come in. Meanwhile, once that amino acid has arrived on the S site, a polypeptide chain or a bond is created between this first codon with the incoming activated amino acid. That bond is a peptide bond that is formed in between there. So once a peptide bond has been formed, then this transfer RNA can detach from this amino acid or can leave the amino acid once the ribosome moves to another codon that is 3 bases ahead in this direction which I earlier showed you. Once the ribosome moves in that direction, Then this empty amino acid or used amino acid is taken to the E site, or is taken out through the E site, and that is the E exit site. Let me write these sites here. This is the A site. This is the P. Which color is that? Let me write here using red. A site. This is the P site, and this is the E site. Okay, I think I've labeled it there. Let me check whether people are capable of seeing what I'm putting there. Let me go back to my meeting. Very good. I see people can see what I can put on the screen, whatever I put on the screen. Okay. So, let me hope the echoes have not come back. So, what was I talking about? Let me not be like a Matia Kashaija. Okay, that's it, by the way. Yeah, so here we are talking about the A, P, and the E sites and how translation occurs. So, Catherine, you have something to say?
Yes, teacher. I'm a bit lost.
Yes. Where can we find you?
Okay. This part of where the light subunit moves 3 bases ahead.
Yes.
That's where I am. So that part of shifting, it's a bit confusing. If you can pardon, please.
The part of shifting.
Yeah. Immediately after the light subunit moving 3 bases ahead.
Okay. Let me use another way of explaining this. Another way of explaining this. Let me explain it in another. Okay, I'll look for it later. All right. Remember what you have to remember. Let me first get here my pointers and what have you. Let me get here the arrow. Yes, here is my arrow, and it is red in color. Should I increase the thickness? Okay, let me just take it. Okay. All right, so after binding of the methionine, rather the tRNA containing methionine on the AUG codon, which we call the start codon, the ribosome, the large subunit together with the small subunits of the ribosome will move. For example, let's take this. If this amino acid is coming, let me get a pen here that I can draw first. This amino acid here is coming to bind on this codon here. Remember, the codon is a triplet base. It's a triplet base. And reading it, one of the properties is that it is a punctuated code, meaning one base cannot be read more than 2 times. So it is punctuated. So this incoming amino acid here, pointer here, this incoming amino acid this one here will bind to these three. Okay, remember the tRNA has the anticodons that are complementary to these codons, so it will bind to these three, and once that has been bound, then the ribosome will move 3 bases ahead, will move in this direction here. So meaning the bases that will be used now will be these ones here, these three. So these three will be shifted to this position here, and these three will allow this incoming amino acid here to bind this second one. The first one, if the first one has already been bound, the second one will come now to bind to these three. Let me draw them in which color? Let me first erase this, and I draw them in another color so that you get it well. Where are my colors? Let me draw it in blue color. So this incoming amino acid here will now bind to these three here, meaning these three will shift to the A site. Then another incoming amino acid which has not been drawn here will come and bind here, meaning at that time the ribosome will have moved to this position here. So the ribosome keeps on moving. Let me get here. This, let me first close it. I don't know whether that is clear now. I don't know whether it's clear. Let me hope people are not jumping out and coming in, because it is already lunchtime. Members, I feel I should stop here because I'm also hungry, meaning the learners are also hungry. Maria Namatov, say something. Your hand is up, Namatov.
Teacher. Excuse me, teacher.
Yes, we can hear you loud and clear.
Teacher.
For me, yes, we can hear you. We can hear you. Please go ahead.
Okay, for me, you've confused me.
How?
Where is that big ribosome moving?
This direction here. The direction where you're seeing this big arrow, which is red, dark red in color.
Okay.
It's moving in that direction.
Okay.
Yes. Is that one clear?
Yes, teacher.
Okay. Any other person? Is that the hen that we shall slaughter during Christmas? There's a hen making noise in the background somewhere. Okay, that's a by the Adikini Petronella, what is she saying? She's offline. Adikini, why are you offline?
No, teacher, I'm online. I don't know.
You're trying to
I'm
press
online.
There's a button you're trying to press, I think.
Button? What button?
You should me here you pressed a button showing that you are offline. Okay, that's a by the way. That is it with the process of protein synthesis. So once you reach this stage where they use this or spent tRNA leaves the ribosome from the E site, This spent tRNA can be taken to the cytoplasm for recycling. Then this amino acid chain, or the polypeptide chain, the growing polypeptide chain, goes through the endoplasmic reticulum to where it is folded into the secondary structure. Then the endoplasmic reticulum encloses this protein in a vesicle, and this vesicle is transported to the Golgi apparatus, where it is fused with the Golgi apparatus for further processing. And during that process, the Golgi apparatus carries out the labeling and addition of the glucose.
Emma, cook the food.
Who is cooking food? Yeah, make sure you share that food with the rest of these members here, because I'm pretty sure they are very hungry and they have not been cooking. They are in the lesson still. Up to still now 3 minutes past 1. We have consumed almost 13 minutes extra. So we are stopping here for now, and I'm giving room for people to talk, unmute and talk and say something, whether you've been benefited or we have confused you more. You also talk to us. If you want to show us your face and your video, you can also show us your face and video, because Adikin is saying I don't think I can enable video because I can share plenty food here. Yeah. You want to send food in soft copy, Adikin? You are not serious, because that food will make us more hungry. The sight of food will stimulate secretion of hydrochloric acid in addition to gas that is present in the gastric juice. So too much hydrochloric acid, if not handled well, will result into ulcers. So let me ask you to unmute and you talk to us, members. As this person is disturbing me in the call, let me first answer.
Music. Adikini.
Guys.
Adikini,
Hello,
spare us.
good afternoon.
Good afternoon.
Guys, excuse me, Amanda. Someone to come help start cooking, because I think I have to just start cooking since there was no one.
Oh my God.
I'm alone here.
Why don't you bring the other one?
As we are cooking, as we study, man, what are you on?
Let me start cooking. I should leave you guys since you guys they helped you cook.
Jayeri.
Naye mwesiimye gwe jjajja we yakufumbidde.
Adikini tukoye muziki wo.
Adikini, we know the vibes.
Adikini will have a home theater.
Vikuyaba, you people have to dance cover night.
Take a video tuzinire mo beti yabadde gatuzinira.
Whoa, wait.
Guys, I think goodbye. We are going to cook. Mweraba.
I want to ban tonight, guys.
Achen.
Wanji.
Guys, I'm leaving.
mambo ya hiyo ni ndani nimewahi kukuambia sitaki kurudia tena. Mambo ya watu kumplete ili kwa huko wakati kidogo kama unalala watu unalala chini kazi kama hakuna sawa. Basi wewe fanya vile ungependa, mimi nilishakuambia. So ukipata tu shida yoyote usiseme sijawahi kuambia. Mimi nitafuta ni nini? I think ndio kitu ya kwanza. Vile vingine vinakuwa rahisi. Baada kama kuna mwingine tena. Ni kama wataandika kwa kutoka sehemu gani. Acha nimalizie hapa hivi.
Alafu hiyo ni nyingine ulinikupe hiyo uangalie.
Kuna mahali nimeandika. Nisomea haraka haraka kuna kitu nataka nimuulize. Application ID nimesema pale tatu alafu HQS